So what influenced your decision in becoming a research scientist in this specific field, and how did you become the laboratory head of neurogenetic disease?
Right. So one of the main reasons I got interested in genetics was because of my biology teacher at school, who was interested in genetics himself. Right? So he had us even at school doing lots of experiments with fruit flies, with Drosophilas, doing crosses and things. So he instilled in us a great interest in genetics. He was also interested in Scottish mosses, which I wasn't interested in at all, but he was obsessed with. So he would have us walking the hills of Scotland trying to find rare mosses, but I didn't enjoy that. I much preferred the genetics. As far as why I got into neurogenetics is concerned, the story that my family tells is that when I was 3, I asked my mum how I could move my fingers when I wanted to. Okay?
Yes.
And so basically where I'm at now is a merging of that question I asked my mum when I was 3 and the influence of the genetics from my biology teacher at school.
Okay. So I can see that you've been working in genetic and muscle research for almost 40 years.
Almost.
And have identified more than 20 genes involved in human diseases, including genetic mutations associated with a group of muscle disorders that appear in birth or in infancy, and Laing distal myopathy. So what has been your most interesting gene discovery, and how has this led you to the current research you are doing?
Okay, when you say it's 40 years, I hadn't quite realised that it is, but it is 40 years. I started doing research in 1976, so next year it'll be 40 years.
Ah, well, yes.
Which is a bit of a thought.
How on?
And I started off studying medicine at Edinburgh University.
Oh, okay.
But I wasn't actually very good at interacting with patients.
Right.
So you did a sort of pre-medical 3 years when you just did the science, and then you started to encounter patients. And when I started to encounter patients, I decided that medicine, being a doctor, wasn't actually for me.
Yeah.
And you could zoom out sideways and do an honours degree, right? And as a medical student, there were 180 students in the medical class, and we had what were called unexaminable lectures.
Okay.
So they weren't lectures that were that you were going to be asked questions on in the exam. So usually somewhere between 10 and 12 out of the 180 students turned up. But those lectures were given by the people in the departments who were actually doing research.
Okay.
So the researchers came and talked to us.
Right.
And one of those guys I found absolutely fascinating what he was talking about.
Oh, okay.
All right? So he was talking about how the nervous system in a developing embryo and muscles get together and talk to each other properly.
Right.
And I just thought that was absolutely fascinating. And when it came to the end of my honours year, he advertised a position as a PhD student, but as a research assistant at the same time. So the position was you were going to be paid a salary to do a PhD, which is really, really, really rare.
Right.
Fantastic opportunity.
Yeah.
And the guy I was doing with my honours with at the time wasn't he. Another guy who, yes, he was into self-stimulating rats, which we could talk about if you wanted, but it's a different story. So I said to him, I really would like to do that PhD. What should I do? So I said, well, go and talk to him. So I went and talked to this guy, and I ended up getting the position.
Oh.
And so I got started working on nerves and muscles in 1976. And so it was what was called a developmental neurobiologist from 76 to 87.
Okay.
And we always felt that what we were doing was going to give insight into these childhood diseases.
Yeah.
But those childhood diseases were genetic.
Yeah.
And in the 1980s, the theory behind finding human disease genes using what was called reverse genetics was put forward.
Okay.
So it became possible to find human disease genes. And an American who was visiting Australia heard me talk about what we were doing with the chicken embryos, and he said, "It's possible to find the genes now, why don't you come and work with me?"
Oh, okay.
So his name was Alan Roses. He was one of the pioneers of doing this gene discovery work. He worked at Duke University in North Carolina. So I got a telethon, right, fellowship for a year to go and work with him and retrained in human genetics. And my job then was to come back here and set up a research lab and a diagnostic lab. So I set up the two different labs. And basically I've been trying to find disease genes ever since.
Yeah.
And the wonderful thing about Australia is that there are amazing families here. So if someone immigrated to Australia, like me with a strange accent, and brought a mutant gene,
yeah,
then there's a big family that we can study.
Yeah.
So these amazing families in Australia we could study. And we could do things that other people in the world couldn't do. So it gave us an old train to making discoveries that others couldn't do.
Right.
The thing that I've enjoyed most doing is our work with skeletal muscle actin diseases. So there are two proteins that are most important in making your muscles contract. One's actin and the other one's myosin.
Okay.
We've actually found diseases associated with both of those. In 1995, I wrote saying that there are probably people somewhere in the world suffering from actin disease, but we just don't know what it looks like.
Yeah.
The year after that, I was at a meeting in the Netherlands, and a German pathologist showed slides of 3 children, two of them had already died, who had this strange accumulation of actin in their muscle fibers. And I said, this might be actin disease. And then we got the DNA sent here. Kristen, who just walked out, she did the analysis of those, found mutations in the actin gene. And then later on, we worked out how it might be possible to treat that. We've shown that it works in mice. So all we have to do now is get that to work in humans. So that's the thing that I've enjoyed most.
Yeah.
Predicting that this disease would exist, finding it ourselves, working out how it could be treated. And we're at the point of trying to produce treatments that you can actually use in the patients.
So in 2008, you edited the first ever book on the human muscle diseases caused by mutations in the proteins of the sarcomere, the functional unit of muscle contraction.
Yes.
So how is this different from your laboratory work, and how has it influenced what you're doing now?
There's a prehistory to that.
Okay.
Okay. So in the early 1990s, when people were just starting to find disease genes, I wrote a grant application which said my hypothesis was that the muscle proteins that everybody knew about, like actin and myosin, tropomyosin and so on, would probably be mutated in some patients who have muscle disease.
Okay.
All right?
Yeah.
That grant application was knocked back on the basis that if my hypothesis was true, the Americans would have found it by now.
Right.
Okay. Which was wrong.
Okay.
They hadn't found it.
Right.
And we were amongst the first to show that those muscle proteins that everybody knew about were in fact mutated in some of these diseases.
Right.
So editing the book in 2008 was basically a statement that in the early 1990s I'd been right.
Right.
Okay?
Yeah.
And all my colleagues and collaborators around the world who've been working in the same area all contributed to the book, and we put it forward.
Oh, I see. Okay.
Right?
Yeah.
And even now, the work we've been doing in the last few years with recessive diseases, which cause unborn babies to become paralyzed before they're born, those are still muscle proteins. Right? But the interesting thing about them is most of those have been muscle proteins that nobody knew existed. when we found the mutations in the gene, it was just a number in the genome, like KLHL40, for example, and nobody really knew what that protein did. Right? And through finding the diseases associated with that gene, other people have now looked at what that protein does in muscle. So it's teaching us more and more about muscle that we never knew existed.
Right.
So that's the crucial thing about that paper. It was a statement.
Yeah.
And it focused people's thinking around the world on that sarcomere structure that everybody thought they knew how it worked.
But,
in fact, they don't. All right.
Right. Okay. So this far down in your line of work, do you still enjoy it?
There are multiple answers to that.
Right.
One is you could ask my wife.
Okay.
How many hours a week I spend doing what I do.
Right.
All right? Which is an awful lot.
Okay.
I'm usually still working after 10 o'clock at night.
Oh, okay.
Most days of the week. I usually come in one day on the weekend.
Ah, okay.
Right? You can't put that amount of effort in unless you really enjoy it.
Yes.
In fact, you might say that you have to be obsessed.
Right. Okay. Yeah.
What I like about what we do is that it's translatable into health outcomes.
Yeah.
So, for example, these muscle diseases that we found genes for are nasty, they're unpleasant. And if a couple have had a child affected by one of those diseases, what they most want is not to have another one.
Yeah.
They don't want another child affected with that.
No.
The only way we can help them do that, avoid having another affected child, is if we find the gene.
Yes.
So, we've had letters, emails, pictures of newborn babies from around the world saying thank you very much.
Oh.
Okay?
Okay.
So, the eventual outcome of what we do is very, very, very rewarding.
That's good. Okay. Yes. And do you know what you're going to be doing in the future with your research?
Yes.
That's good.
You need to be strategic about what you're hoping to do. So there's still a whole lot of disease genes to find. The Western Australian Department of Health, interestingly, is a foundation partner of the International Rare Disease Research Consortium. And the aim of that consortium is to find every Mendelian disease gene by 2020. Okay?
Right.
Now, in 1986, basically, no Mendelian disease genes were known. So between 86 and 2020 is how many years?
Did you say 96?
86.
Oh, 86. 34?
34 years. So in 34 years of human history, we may have gone from knowing absolutely nothing, basically, to knowing every human Mendelian disease gene.
Oh, okay.
Right. So in my working life,
Right.
I'm hoping still to be working in 2020. Right?
I'm sure you will be.
There's no guarantee. Yeah. So that's one thing.
Yeah.
And it's called the diseaseome.
Okay.
So we will have completed the diseaseome for Mendelian diseases by 2020, or near enough to make no difference. And that's a fantastic achievement. You know, my group here will play a part in that, in the particular diseases area that we're interested in. So that's one thing. The next question is, what does society, what will your generation do with that knowledge? Right? Because knowledge is power.
Yes.
So knowing that gives future generations power over their genetics.
Yeah.
So in a review I wrote in 2012, I said when any couple has a child, they play genetic roulette, because each of us does not know what nasty genetic diseases we're carrying.
No.
Although the estimate is each of us is carrying between 3 to 5 genetically lethal diseases.
Ah.
So if you have children with someone who's also carrying
Yeah.
one of those 3 to 5, 1 in 4 of your children is going to have a really nasty disease.
Ah.
So one of the things I've been talking about for a long time, since the early 1990s, and a lot of other people have as well, is wouldn't it be a good idea if we knew what nasty recessive genetic diseases we were carrying
Yeah.
before we decided to have kids?
Yeah. Definitely.
So what? Well, you say definitely, you see, but other people might not think that way.
No.
Okay?
They might.
It's a very tricky ethical decision.
Okay.
All right? So that's an area that I'm really interested in.
Okay.
And it's called preconception carrier screening. And it's something I'll be devoting a lot of time to over the next 5 years.